Hi. So we'll continue with the gene regulation part. So we'll start with gene regulation in prokaryotes. The first part and most important regulatory mechanism that you have to know is Lac Operon. Okay. So this was described by Jacob and Monod. So we'll just look into what exactly an operon is. In our previous section, we have seen that operon is genes involved in a metabolic pathway are sequentially arranged are sequentially arranged on an mRNA. First point. Second point is it is regulated by a single promoter or a regulatory region. For example, say glycolysis. Only example. Okay. Glycolysis. We know that the enzymes are hexokinase, then phosphofructokinase, then pyruvate kinase, all this. Now, all these enzymes will be arranged sequentially. Hexokinase, phosphohexose isomerase, phosphofructokinase, aldolase, so on. Okay. All these enzymes like say pyruvate kinase, all these enzymes belong to one metabolic pathway only. Same pathway. This is the specialty of prokaryotes. This doesn't occur in eukaryotes. In eukaryotes, we have seen that all of these are dispersed. Whereas in prokaryotes, all these enzymes of a metabolic pathway, they are present in sequence and they have a regulatory sequence before that. This regulatory sequence and these genes which code for different enzymes of the same pathway, this is called as an operon. Okay. So what is there inside an operon? Enzymes of a same pathway are in sequentially in a linear order regulated by a regulatory sequence. Okay. So that is an operon. Now, Lac Operon is an operon for lactose metabolism. What is happening to lactose? Lactose has to break down to form galactose and glucose. The enzyme which is required for this reaction is beta galactosidase. Then there is another enzyme permease which allows influx of lactose inside the cell. Then there is another enzyme which acetylates this lactose, that is called as thiogalactoside transacylase, which acetylates this lactose. So this metabolism of lactose, it requires three enzymes: beta galactosidase, a permease, and thiogalactoside transacylase. Three enzymes are required. Okay, so that is the metabolism. Now this metabolism occurs inside the bacteria. Whenever lactose is present, lactose is broken down to galactose and glucose. Now let us see how this metabolism is regulated at the transcription level. Okay, so what is happening? Let us look at the operon first. Okay, so there is an mRNA. This mRNA, it has 4 genes. Okay, this Lac Operon has 4 genes. First one is Lac I, which produces the repressor synthesis or transcription. Okay, then there is Lac Z, which is for beta galactosidase. This is here Lac I, this is here Lac Z. Okay, next is here Lac Y for permease and Lac A for thiogalactoside transacylase. So here we have a Lac Y and we have a Lac A. Okay, apart from these 4 genes, there is one operator region wherein the repressor binds. Repressor is an inhibitor. We have seen in the introduction class what is a repressor, what is derepression and all that. Operator, it is a place where the repressor binds. Okay, then it also has a promoter region. In our transcription class, we have seen a promoter region is a place where your RNA polymerase will bind. Here RNA polymerase will bind. Then we have a cyclic AMP response element. Cyclic AMP response element called as CRE, wherein the cap cyclic AMP will together bind. What is this cap? This cap is catabolite gene activator protein. So many terminologies we'll see. Okay, so okay, we are having an operator, we are having a promoter, and we are having a cyclic AMP response element. What was Lac A doing? Lac A, it produced thio. Okay, Lac A. What did it do? It produced thiogalactoside transacylase. Okay, Lac Y produced permease. Lac Z produced beta galactosidase. Okay, operator is the region where your repressor protein will bind. Repressor protein will bind. Promoter is an area where your RNA polymerase will bind. RNA polymerase. CRE or cyclic AMP response element is a place where the cap protein along with the cyclic AMP will bind. And Lac I is the region which produces or which transcribes for the protein repressors, for the 4 subunits of the repressor protein. These 4 subunits of the repressor protein, they will form a complex, and this complex will come and bind on the operator region, so that the operator region is always blocked. Okay, the operator region is always blocked. This Lac I expression is always constitutive. Constitutive always present. It keeps on producing this repressor protein which will form a complex, and this complex will keep on obliterating the operator. Okay, so this entire thing is an Lac Operon. This entire thing is a Lac Operon. This entire thing is a Lac Operon. What does it contain? 4 genes: Lac I, Lac Z, Lac Y, and Lac A. Okay, an operator region where the repressor binds, a promoter region where the RNA polymerase will bind. What is the function of RNA polymerase? To transcribe. What it will transcribe? It will transcribe these 3 enzymes: beta galactosidase, permease, thiogalactosidase, transacylase. Okay. Then there is a cyclic AMP response element which will bind with cap and cyclic AMP. This is absolutely essential. This is absolutely essential for the RNA polymerase to bind to the promoter region. Without cyclic AMP or cap, RNA polymerase will not bind to the promoter region. Okay. Then Lac I is the region in the DNA which transcribes proteins, repressor proteins. These repressor proteins will form a complex, and they will bind to the operator region by 2 subunits only. All 4 subunits won't bind, only 2 subunits will bind to this. Okay. Now, whenever there is a repressor protein, that means there is always a block and RNA polymerase is not going ahead. Okay. Now we will see how this repression is removed by a process called as derepression. So Lac Operon is basically a model of derepression. Okay, Lac Operon is basically a model of derepression. What is an operon? Genes involved in a metabolic pathway which are sequentially arranged on an mRNA. This particularly occurs in prokaryotes. There are so many operons like that. We are having Lac Operon, we are having tryptophan operon, we are having arabinose operon, which we'll be seeing in our subsequent videos. Alright. So basically, if you remember the pathway of glycolysis, hexokinase, phosphoexoisomerase, then phosphofructokinase, aldolase, glyceraldehyde 3-phosphodehydrogenase, all those enzymes will be sequentially arranged inside the mRNA. Before that sequence, there will be a regulatory element. So this regulatory sequence, along with the genes arranged in array, it is called as an operon. So now we are studying a Lac Operon, which is for lactose metabolism. Lactose metabolism generally in bacteria is lactose is broken down to glucose and galactose by beta galactosidase. Lactose is permeated inside the cell or is pulled inside the cell with the help of permease. Lactose, before getting converted to galactose and glucose, needs to get acetylated, which is acetylated by thiogalactoside transacylase. Now, these 3 enzymes are transcribed from a single mRNA. They are arranged in sequence as in Lac Z, Lac Y, and Lac A. These 3 genes of a single pathway are arranged in sequence, which is controlled by regulatory sequences. These all are regulatory sequences: operator, promoter, CRE, Lac I, all these are regulatory sequences. Okay. Now, whenever lactose is present, RNA polymerase here will bind to it and it will undergo transcription. We'll see. So a promoter region, then an operator region, and three genes. All right. Here we have seen a basic structure of Lac Operon. How these enzyme genes for different enzymes are arranged in sequence: Lac Z, Lac Y, and Lac A, and it is followed by, or it is preceded by, their regulatory elements. Okay, so that is a concept of Lac Operon. Now let us see how this Lac Operon works. Let's understand a basic concept first before going in depth. Okay, so first let us imagine that lactose has come inside the cell. Some lactose has come inside the cell. Okay, now let us imagine that these dumbbells are lactose, 1 unit of beta galactose and glucose. Okay, galactose and glucose. This is a lactose molecule. This lactose, whenever it comes, we have seen that on the operator side, Lac I, this will always, Lac I has Lac I is a housekeeping gene, housekeeping or a constitutive gene, which always produces repressors. Okay, these are repressor molecules. These repressor molecules, they form a complex and they will come and bind to Lac O. They'll come and bind to Lac O, and the RNA polymerase which is bound here to the promoter site, it cannot do the transcription normally. So what happens is, when these lactose molecules will come, what they will do is they will bind with these operator molecules. So these repressor molecules, and these repressor molecules will come out of the operator site, and there will be a structural change in them. So that they cannot bind to the operator site again. Okay, so now what happens, whenever lactose comes inside the cell, this will go and bind with the repressor molecules, and repressor molecules are removed from the Lac O site. When these repressor molecules are removed, what happens is polymerase will start, RNA polymerase will start the transcription, and Lac Z will form beta galactosides, Lac Y will form permease, and Lac A, it will form thiogalactoside transacylase. So what is happening basically, lactose when it comes inside the medium, it will remove the repressors which were present here. When these repressors are removed, what will happen? RNA polymerase will produce the 3 enzymes. Now here lactose becomes the inducer. Okay, so whenever lactose is present, whenever the regulator is present, there is increased transcription. Whenever regulator is present, there is increased transcription. So this is an example of positive regulation. This is an example of positive regulation. Okay. Now, if lactose is the inducer, then there are other molecules like isopropyl thiogalactoside thiogalactoside or IPTG, isopropyl thiogalactoside, which can also work as an inducer. It can also work as an inducer, though it is not a substrate. It is not a substrate, but still can induce this transcription. That is why this is called as gratuitous inducer. It is called as gratuitous inducer. Gratuitous inducer. Which is one? This is the one. Gratuitous inducer, isopropyl thiogalactoside. It is not a lactose is the substrate. Lactose is the substrate. This substrate is inducing the RNA transcription. The transcription for its enzymes is understood. Okay, that is an inducer. But a non-substrate which is also inducing is called as a gratuitous inducer. Example is isopropyl thiogalactoside. So basically, first repressor molecules are bound here. They are constitutive. They are always present. Whenever lactose comes inside the cell, this lactose will come inside and it will remove this repressor molecules. That is, it causes de-repression. It removes the repressor by changing their structure, by binding with them and changing their structure. De-repression, it causes de-repression. So once de-repression occurs, what happens is RNA polymerase is free to transcribe the genes for beta galactosidase, permease, thiogalactosidase. These RNAs that are formed now, these RNAs that are formed, that will go inside that will go onto the ribosomes and they will synthesize the protein beta galactosidase, permease, and thiogalactoside transacylase. Now, not just the substrate, but also other substances like isopropyl thiogalactoside can also induce this change. That is why it is called as a gratuitous inducer. Okay. Now, not only lactose, but bacteria can also use other substances like, say, glucose. Glucose is the principal. It is the principal substrate. Whenever glucose is there, bacteria will not utilize lactose. Whenever glucose is there, bacteria will not utilize lactose. Arey bhai, if biryani is there, why will anyone eat dal chawal? Correct. So glucose is the biryani for the bacterial cell. So whenever glucose is present, the first preference for the bacterial cell will be glucose. It will not take lactose. Now this presence of glucose will inhibit this operon. It will negatively inhibit this operon. Now we will see how that happens. Basic concept, is it understood? Clear? Three genes, LAC-Z, LAC-Y, LAC-I, which are transcribed into the mRNAs for beta galactosidase, permease, thiogalactoside transacylase. These will undergo translation to produce the protein beta galactosidase, permease, and thiogalactoside transacylase. RNA polymerase is inhibited because first there is a repressor molecules. That is why RNA polymerase cannot synthesize this. Once lactose comes inside, it will bind to the repressor molecules, remove the repressor molecules away, and RNA polymerase will synthesize these three things. Okay. Now, let us go into depth and see what happens in three different scenarios. Okay. Okay. Let us look at the first scenario, wherein First scenario wherein lactose is present or isopropyl thiogalactoside is present or and glucose is absent or glycerol is absent. Glucose and glycerol will have the same effect on the regulation of Lac Operon. Okay, so first what happens is lactose has come inside the cell. Some amount of lactose has come inside the cell. Okay, so this is your lactose. This lactose, what it does on the operator region, we are having 4 repressor molecules already. So this will go and bind with this repressor molecules and it will convert them it will change the shape. All right, of these molecules, and the complex is no more stable, and the complex now comes out of this. Okay, whereas these repressor molecules come from, they have come from expression of lac I, which has formed a complex. This complex had attached to the operator molecule and it was there. So whenever lactose has come inside, lactose comes there and it will remove this operator and the operator site is now free. Whenever there is low glucose, an enzyme adenylyl cyclase is activated. This activated adenylyl cyclase, it will produce increased amount of cyclic AMP. This increased amount of cyclic AMP will bind with the cap protein. Okay, and this cap protein will come and bind here along with the cyclic AMP. Now this cap and cyclic AMP, these are the stimulators for your RNA polymerase to bind here. This is the positive stimulus. Okay, so now RNA polymerase is bindas. It has no obstruction. First obstruction that was there, that is your repressor molecules. These repressor molecules are removed by lactose. Okay, the second thing that was required was cyclic AMP and the cap protein to bring the RNA polymerase and attach it to the promoter. Okay, so two have occurred. Why? Because there is no glucose, low glucose amount. Whenever there is low glucose, adenylyl cyclase will get activated, which will produce a lot of cyclic AMP. This cyclic AMP will bind with the catabolite gene activator protein. This cap complex, it will bind to the cyclic AMP regulatory element or response element, which will activate or it will recruit the RNA polymerase to the promoter. This RNA polymerase, once it attaches to the promoter, what happens? The repressor molecule is no more left. This repressor is gone, and now RNA polymerase will start synthesizing the RNA for LAC Z, LAC Y and LAC A. So RNA polymerase now becomes free and it will move ahead. It will transcribe the RNA for Lac C, Lac Y, and Lac A. Okay, so these will go to the ribosomes. On the ribosomes, they will produce the protein. On the ribosomes, they will produce the protein. What is this protein? Beta galactosidase, permease, and thiogalactoside transacylase. Okay, these three are produced. So two important things are required for this. First important thing that you need to know is the repressor molecule should be removed for the Lac Operon to act. Second, there should be a cyclic AMP and cap for the recruitment of RNA polymerase at the promoter site. So this is the second thing. These two things are absolutely essential for the perfect or for the correct running of this Lac Operon. What was there? Lactose had come. This lactose has bound to the repressor molecules and it has changed their conformation and released them from the operator. So the operator site is now free. When there is low glucose, there is increased adenylyl cyclase, which will increase the cyclic AMP, which will bind with the cap protein, and this cap cyclic AMP complex, it will bind to the cyclic AMP response element, which will recruit the RNA polymerase. This RNA polymerase will cause the transcription of the three genes Lac Z, Lac Y, and Lac A, and this transcription will finally lead to translation of the enzyme beta galactosidase permease and thiogalactoside transacylase. Now let us look at the second scenario wherein lactose or isopropyl thiogalactoside is present as well as glucose and glycerol, they are also present. What will happen now? See, if glucose and glycerol is present, what happens is there is decreased adenylyl cyclase. Decreased adenylyl cyclase, which means there is decreased cyclic AMP, which means there is no binding of cap and cyclic AMP. Why? Because there is no cyclic AMP to bind with cap. If there is no cyclic AMP, then this space is vacant. Okay, and this space was our second point, which was absolutely essential for the RNA polymerase to come and bind. Here. Okay, second thing was the operator. What happens here? Because lactose is present, this lactose, it will go and bind with the repressor molecules and it will release these repressor molecules. Which were bound here. It will release. So what happens is, in the presence of lactose, the operator site is free. In the presence of lactose, the operator site is free. So RNA polymerase should be able to transcribe. But what happens is the on switch or the recruiter of RNA polymerase, that is this cyclic AMP cap, is absent. The second most important requirement for the transcription is absent, though one is present. What was one? Removal of the repressor molecule. That happened because of the presence of lactose. But the second one, that is this recruitment of RNA polymerase, which requires cyclic AMP and cap complex, that is absent. Because of this, RNA polymerase will not come and bind, or even if it can bind, it binds very little and the transcription is inefficient. So the transcription doesn't occur. Okay, so whenever glucose is present, that is why body will utilize glucose and it will not utilize lactose. Why? One of the reason for that is there is decreased adenylyl cyclase in presence of glucose, which means decreased cyclic AMP levels. This cyclic AMP will not bind with cap, and this cap cyclic AMP will not be able to recruit the RNA polymerase for the job to be done, though the operator site is vacant. Okay, now let us look at the third scenario wherein lactose is absent and glucose is present. Okay, third scenario, there is no lactose but glucose is present. When lactose is not there, what happens to the repressor molecule? Lac-I is producing this repressor molecule which forms a complex, and this complex goes and binds to the operator. This is housekeeping. So always repressor is present, always present. Okay, so this repressor molecule is present. All right, now lactose is not there, so operator site will not be vacant. When operator site is not vacant, there is no chance that RNA polymerase can go further. RNA polymerase cannot go further, not possible. And because glucose is present in the medium, what will happen? The adenylyl cyclase levels are low. Adenylyl cyclase levels are low. That means low CAMP levels. That means there is no binding with CAP. When there is no binding with CAP, there is no recruitment of RNA polymerase. So the first requirement and the second requirement both are not happening here. All right. So by these three scenarios, we can understand one thing is lactose should be present and it is a positive regulator. Okay, how it is regulating? By derepression. For the Lac Operon to act, glucose levels should be low. Glucose levels should be low. How that is helping? It will increase the adenylyl cyclase, which will increase the cyclic AMP, and this cyclic AMP will bind with CAP. This will bring about RNA polymerase action. This RNA polymerase will synthesize the three enzymes required for lactose metabolism. What are they? Beta galactosidase, permease, and thiogalactoside transacylase. Okay, so that was about Lac Operon. All right.